Okay, MCAT enzyme kinetics with a table of data. Which of the following is most consistent with the mutants in the glutamate codon and hexokinase? You're given 4 answer choices as you see there. You have mutant 1 and mutant 2. You have 2 choices that affect a lysine, 2 that affect the aspartic acid, 2 that affect substrate binding, and 2 that are mutants at the active site. So what do? So you see the data, and only 3 of the entries matter. That is the hexokinase 1, the mutant hexokinase number 1, and the mutant hexokinase number 2, and you're given KCATs and KMs. So when you see tables like this, look for the data that is similar as well as different, and when we do that, we see the KM values for wild-type hexokinase 1 and the 2 mutants are pretty similar. So what does that mean? Well, let's think about what KM means. It means the concentration of substrate at 1/2 Vmax, okay? It also refers to the inverse of the binding affinity. For example, if you have a high KM, you have a low affinity for the substrate and vice versa. So therefore, if you take a look at the answer choices, what you would think is that it doesn't affect the substrate binding, and so we can eliminate choices A and D and now focus our attention on the KCAT values. What you see for the choices B and C is that we're discussing a aspartate at the active site where the normal residue is glutamate. And if we compare aspartate and glutamate, they both have negative charge. The only difference is one methylene group. So if you can look and think about hexokinase 1 mutant number 2, it has almost half of the activity as hexokinase 1 does, but mutant number 1 is far much reduced. So the most parsimonious answer is to think that number 2 is mutated to an aspartate at the active site because it still contains a significant amount of enzymatic activity. Come check us out at medpathway.com.